Remember the characteristic of life, that living things are organized. Now, we're really focusing in on atoms bonding together to make molecules, molecules coming together to make a cell, and cells making tissues, tissues working together to make an organ, and all the organs interacting and communicating with each other to make organ systems, and all of those organ systems being contained within one individual organism. So in the beginning part of the semester, we do like to just briefly touch on some basic concepts from chemistry. This will help us to understand a little bit more about how we get to a cell, that smallest unit of life, and then we take for granted that cells and life have happened, and we talk about all the different ways that cells and life have changed. So let's take a step back and kind of ground ourselves in some concepts of physics, some concepts of geology, astronomy, and some concepts of chemistry. So when we look at the origins of the universe, the theory that helps us to understand the origins of the universe is the Big Bang Theory. Now, we're not going to get into too much detail. What we do want to know is that we have atoms, and lots of these atoms were all moving about. Now, the 6 atoms that we find in all living things will be carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Of course, there are a lot more atoms. But in order to understand how these atoms moving about, bumping into one another, might have started to have and make bonds, we do have to understand a little bit about the structure of an atom. So when we look at the structure of an atom, we're going to have protons. Protons are the subatomic particles that are positively charged. So I'm just going to draw one happy little proton here. We're going to have little neutrons, and neutrons do not have a charge. And then we are going to have the most important subatomic particle, at least from a biological point of view, and that's going to be the electrons. I'm going to draw a little electron out here, and I'm drawing my electron in red. Now, our electron is going to be in something called an electron shell. An electron shell is just where we can find the electron. Now, electrons are constantly moving. They're always wiggling and jiggling. They never sit still. And what happens is when we have the nucleus of our atom with our protons and neutrons, our electron is going to spin around this nucleus, but it's going to do so in a 3-dimensional space. Now, I can't show that on my board, which is why I drew this kind of, should be circular shaped, but why I drew this red line out here outside of my nucleus. Because what I want to do is I want to represent the place where that electron will be. Now, the reason why the electron shell is important is because how many electrons an atom has in its outermost electron shell is going to determine whether or not that atom wants to make bonds. So in this example, what I have drawn or what I have modeled here, this is the hydrogen atom. And we're going to abbreviate the hydrogen atom with just the letter H. And it just has one little old electron here. It has one electron shell. Now, this electron shell would be full if we had 2 electrons in it. We have just 1 electron. So this electron shell for hydrogen is not full. And hydrogen really, really, really loves to make bonds. So I'm going to draw another atom just over here to the side. And for this atom is going to be helium. And helium we're going to abbreviate with He. And helium is really neat because it has 2 electrons in its outermost electron shell. So the thing with helium is that helium has a full outer electron shell. It doesn't want to make a whole lot of bonds. Now, keep hydrogen and helium in your mind when we get a little bit later on in this lecture. This outer electron shell is going to help us a little bit more, understand a little bit more about hydrogen and helium and some other interesting things. So, we look at an atom. the most important part is going to be the electrons. If the outer shell is not full of electrons, that atom is going to want to make a bond. So we have the Big Bang. We have all these atoms moving around and bumping into one another. We find that the atoms that had unfilled outer electron shells started to make bonds. Well, when those atoms started to come together to make molecules by bonding, then they were more likely to bond with other atoms and bring more things in and more things in. And now we've got to start to talk about the formation of planets. So with the formation of planets, we find that we go from this sort of amorphous nebula with a bunch of atoms and molecules bumping into one another, starting to collectively glob together. And if we get the right type of atoms all starting to move together and starting to bond together, and we get these larger units, what we can start to see is a gravitational force being exerted on the surrounding area. Now, this aspect of a gravitational force is what defines a planet. So when we look in our own solar system, what we find is that poor little Pluto out there, it's just a protoplanet. It doesn't have enough gravitational force to dominate the area around it. But all the rest of the planets, including Earth, have that gravitational force to dominate the surrounding area. So now let's take a little bit closer look at our own solar system and one of the most important aspects, which is the sun. Now, the sun is really super unique. With technological advancements, we now know more about the sun than we ever have before, and every time we make new observations, it kind of blows our mind because there's so many interesting things going on with the sun. So the sun is a perfect sphere. It has a lot of magnetic fields. You're going to see in the lecture slides that there are these weird tornadoes that happen, which are super spooky. And we've got visual now of the nuclear fusion happening. Hydrogen and helium coming together. And through that nuclear fusion reaction, we get a release of a tremendous amount of energy. That energy is what we need here on Earth. Now, our simplest definition of energy is the capacity to do work. All life on this planet is transforming energy. And most of that energy originates from the sun. The plants and organisms that can do photosynthesis are transforming that solar energy into essentially sugar molecules, glucose molecules. And then organisms like you and me and cats and dogs and wolves and elk, we all eat and chew and consume those glucose molecules so that our cells inside of our body, transforming energy, can stay alive, maintain homeostasis, and demonstrate the rest of the characteristics of life. So this is why we talk about the sun, and why I wanted to take a moment and just in the lecture slides go, wow, the sun is truly amazing. It's got a lot of amazing properties. Now, we want to stay focused on life on Earth. When we look at life on Earth, we know that the Earth consists of multiple different layers. And those layers end up being really unique and is part of the reason why life could originate here and also why life can continue to be here. When scientists are looking elsewhere in the universe, they're looking for a lot of these different characteristics that we find in our own planetary composition, in the way how it's able to exert the gravitational force, in that core, mantle, crust, atmosphere relationship. And we look to Mars a lot of times because Mars has a lot of the same characteristics, but it lost its atmosphere. And that's really significant. So we do that comparison of like, whoa. If you lose an atmosphere, if you lose that layer on a planet, you may not be able to have life. So there are scientists who are hoping that they can find life. There has been some evidence that indicates that probably life did exist on Mars. And I can't wait till the day when we get more information back and we get to know more about what that might have been, what that might have looked like. And the other weird one is Venus. Now, I know Venus is super hot. Its atmosphere, at least from our point of view, is super toxic. But there are some researchers that have some evidence that maybe floating around in that really toxic atmosphere, there could be evidence of life. Well, see, already we start to get down a really sticky path, and we'd have to get into a lot more details. So let's stay focused on life on Earth here. Turns out life hasn't been existing here very long. Our planet spent a long time in this very turbulent time when the crust was very molten. We had a lot of meteorites bombarding it, and it was just a tough time. But as the Earth started to cool, we started to get the formation of crust. We get those great big supercontinents. We get Pangaea, and then we get Gondwanaland. And some of the important things about this is that this shift, this movement of the chunks of crust. Now, we take for granted this concept of plate tectonics. The crust is broken into chunks, and those chunks are constantly moving. But when scientists first started discovering fossils in very different continents, but they were the same fossils, well, this kind of flew in the face of the paradigm that the crust is one solid unit and was never broken into chunks. It has always looked this way. So this is one of the first times when scientists were really starting to challenge our understanding of the crust, understanding of our planet. And now we take for granted continental drift and plate tectonics. When we talk about continental drift, please, always keep in mind how slowly this happens. The speed of our crust moving is exactly what allows species and life to stay the same for a long period of time. But then what we see is we see these dramatic shifts. We see these dramatic speciation events. We see these dramatic adaptations occur alongside that sort of threshold being crossed, that moment when one continent is finally far enough away from another, or is crossed into a different place where we have changes of temperature and precipitation, or a different exposure to ocean currents, and that affects all of the life. So across our planet, we know that things stay relatively the same for a long period of time, and that we go through these shifts and changes that can be relatively dramatic. So keep in mind the speed of continental drift. This will be one of those themes that keeps coming up throughout the rest of the semester. So after looking at the origin of our planet, we do want to take a look at some of those big moments in time. We want to look for that moment in time. We have the origin of the first cell. We have the evidence of those stromatolites, those huge bacterial mats, all of the different layers representing bacterial cells being laid down over time like the rings of a tree. So the further down into that stromatolite you are, the older the bacterial cells, and the closer to the top, the younger those bacterial cells are. We want to look for the origin of the first photosynthesizing cells. And the reason why this is important is because photosynthesis, with its release of oxygen, is what shifts the Earth's atmosphere. So the Earth's atmosphere, the second atmosphere, was high in methane. And methane was what some of the first cells, those first bacterial cells, were consuming. They were essentially eating or breathing in the methane. The group, or this potentially kingdom of species that were doing this, were called the methanogens. Some of those species still exist today. They can't survive in an environment with a lot of oxygen. So when cells started doing photosynthesis, capturing sunlight energy to make sugars, the byproduct of that was oxygen. And as the levels of oxygen increased in our atmosphere, we had our first mass extinction of those methanogens. They couldn't survive as the oxygen levels were starting to increase. Now, this seems very strange because the life that we're familiar with, that we've been observing, well, that's based on a high oxygen composition in our atmosphere. Now, weirdly enough, our atmosphere is only about 20 to 21% oxygen. Most of our atmosphere is composed of nitrogen. So that's kind of an interesting shift that we see in our atmosphere. But it's this balance of gases in the atmosphere that has allowed our life to be the way that we see it today. And one of the interesting experiments that we see happening is how our activity, shifting the composition of carbon dioxide and methane in our atmosphere, is starting to affect life here on the planet. So when we talk about climate change is the changes in predictable patterns of temperature and precipitation. We know that this is linked to the greenhouse gases, and two of those greenhouse gases that are trapping heat closer to the surface of the planet are carbon dioxide and, more importantly, methane. And it turns out methane is a much better gas for trapping heat here near the surface of the planet. That trapped heat is what's starting to shift our predictable patterns of temperature and precipitation. The thing that I personally watch and have been watching change over time is this wobble in the jet stream. Now, the wobble in the jet stream ends up being really crucial, as that jet stream is circulating around our planet, and at least here in the Midwest, it has a pretty regular cycle. But when it starts to wobble, and it's a little bit closer to Russia, or if it starts to wobble and sink a little bit here on North America, we see that pulling of polar air, either pushing it closer to Russia or pulling it closer here to us in the Midwest. And in 2019, you may remember the polar vortex and that extreme cold that we experienced as that polar air got pulled and sucked down to our region. Now, interestingly, we've had these similar types of polar vortexes affecting us in the summer. So in August of 2019, we had this really kind of couple of weeks of just very, very, very chilly air, and that was really unusual. That was the polar air shifting our way. But in August, we don't really complain that much if the highs are only in the 50s. We're like, whoa, this is really kind of nice and refreshing. But if we look at that larger scale pattern, this is hugely disturbing. It should never get that chilly in August. And currently what's happening here in January of 2021 is that polar air up in the polar regions has split into two larger chunks. This has happened a couple of times in the last 20 years and has led to very, very extreme weather events. Now, right now, scientists are not predicting a huge weather event here in the U.S. They don't actually know how the splitting of the polar air is going to affect us, but they do know that polar air is probably going to have, or they predict that polar air splitting is going to have huge effects over through Europe. So we'll see how this works out. Regardless, it's very scary. I know a few years the jet stream that hangs out kind of close to the equator did this really creepy thing that nobody had ever observed before. It dipped below the equator. Scientists were able to watch it happening in real time and then go, wow, Doesn't seem to have caused a lot of huge things, but scientists are worried because all of this changing will have an effect on life and is already having an effect on life on this planet. So the big takeaway here is that every single layer affects life. The life that we see now is the result of all of the changes that have happened in the past, and that whatever is happening now will affect life going forward into the future. And we have to remove our values. If you are deeply passionate about something like polar bears, it's kind of sad because these changes could mean that your polar bears are going to go extinct, and that's kind of heartbreaking, but that's a value. The planet doesn't care. The Earth is going to keep spinning. It's going to keep circulating around the sun. It's still going to have the same relationship that it's always had with the moon. Until the sun decides to explode, everything is going to be fine from the planet's point of view. But from the human point of view, we may want our species to continue to exist with a certain standard of living, certain things that we have grown accustomed to, certain species that we value. So that's where we always have to go, here are my values, but here's the way how the system is actually working. And evolutionary biology, sometimes it gets really hard to separate those two. And I hope that throughout this semester, you are able to separate how you feel about something versus what is actually happening in front of you. So getting back to a little bit of geology, let's talk rocks. Now when we look at the rocks, the reason why we're touching on the different types of rock is really so that we can understand more about our crust and specifically understand more about sedimentary rock. Sedimentary rock is formed through the process of erosion. Erosion happens because of water. And water is such a hugely powerful molecule. So let's stop for just a moment and look at the structure of water so we can understand more about why it can be so powerful, why it can do erosion, why it can be the crucial molecule in helping to make sedimentary rock. So water is going to be 2 hydrogen atoms and an oxygen atom bonded together. So here I'm going to draw my little hydrogens. And if you remember, hydrogen just has 1 electron. So hydrogen has an unfilled outer electron shell. It wants to bond. Oxygen, in its outer electron shell, it has an unfilled outer electron shell as well. So what's going to happen as soon as I get oxygen close to hydrogen is oxygen and hydrogen are going to want to make a bond. Now, when we draw and show this bond, we often show it as this little cone. And the reason why we do this is because we want to communicate the idea that the electron from hydrogen is going to spend more time hanging out here around the oxygen's nucleus. So when 2 hydrogens bond with oxygen, their electrons spend more time hanging out here around the oxygen nucleus. Because oxygen has more electrons, that's going to make this side of our molecule more negatively charged. When hydrogen's electrons are spending more time hanging out down here, that's going to make our hydrogen side more positively charged. And so we get this difference in charge on either side of our water molecule. When we have a difference in charge across a molecule, we call our molecule polar. So water is a polar molecule. This unequal sharing of electrons between hydrogen and oxygen is called a covalent bond. So because of the covalent bond between hydrogen and oxygen, it causes our water molecule to be polar. It is the polarity of water that gives it all of those beautiful properties that are in the lecture slides, and it's why erosion can happen. Water is a solvent. It can break the bonds of all kinds of other molecules. So let's say we take some salt. Salt is sodium and chlorine bonded together. We put that salt into some water. What's going to happen is water is going to be able to break the bonds of the salt molecule. It's going to grab a hold of the positively charged sodium and tear it away from the negatively charged chlorine. And it's just going to get in there like the most destructive third wheel in a relationship and just break everything apart. Water can just break the bonds of things. So when we're talking about erosion, really what we're talking about is this capability of water, this polar molecule, because of this covalent bond, to be able to break the bonds of other molecules. And then water grabs a hold of those other atoms and other molecules and goes, come with me, come with me, come with me, come with me. Because water moves across the landscape, it's going to be taking all those molecules with it. Water will start to collect into creeks and streams and rivers and lakes, into larger and larger bodies. Water likes to be with other water molecules. Water is cohesive. So, when water is moving across the landscape and water molecules are grabbing onto each other and grabbing onto other molecules and making creeks and rivers and streams and lakes and bigger bodies of water, what we are going to find is that those molecules are going to do something, we call it falling out of the water column. So as water molecules start to collect into larger bodies of water, they'll start to let go of the other molecules that they've drug with them. So we get this predictable rate, this predictable formation of sediment, other molecules accumulating at the bottom of bodies of water. If we have lots of little tiny molecules, those molecules are going to move further, whether it's through a river or a lake or a pond. All small molecules can go farther. Larger molecules are going to fall to the bottom a lot sooner. Yay, gravity. If we have really fast-moving water can move all molecules much farther. If we have slower-moving water, then we're going to find the accumulation of sediment happen a lot sooner. Now, fortunately, we don't have to do the calculations to figure out the rate of sediment formation. But I can tell you that scientists and researchers and engineers, they've done these calculations. For every body of water, for every amount of rainfall, they know the rate of sedimentation across our entire planet. It's actually quite beautiful and quite brilliant, but the calculations are brutal. As predictability is what we get to manipulate in evolutionary biology. So if we're looking at sedimentary rock, all of those layers of sediment made by erosion, made by the bodies of water and the particles falling out, then what we know is that if we go deeper down through that column of sedimentary rock, that the layers near the bottom are older and that the layers near the top are younger. When species die and their bodies become mineralized in the sedimentary rock, essentially when those living organisms become fossils, we know that fossils that are found down lower in the layers are older and those near the top are younger. So this gets us to the law of superposition, which essentially is using sedimentary rock to get an approximate age of fossils based on these layers, older near the bottom, younger near the top. So if our geological feature is like the Front Range of the Rocky Mountains, where we see that all of those layers have been tipped sideways, we can still look through the layers like looking through a slice of cake and know that those on the bottom versus are older than those at the top, even if our layers have been shifted in a different direction. So this has happened in several places across our planet. And I bring up the Front Range of Colorado because some of the fossil discoveries of mammals are coming out of that region, and it's absolutely brilliant to know more about the mammal branch on the phylogenetic tree. We'll be touching base with this throughout this semester, and we'll see this law of superposition come up or be an underlying assumption when we're reading about case studies and conclusions throughout this semester. Now, using sedimentary rock and getting an approximate age is nice. If we want a more accurate age of a fossil, then we can use isotopes. And isotopes are atoms that have more neutrons in the nucleus. And with an isotope, we get a predictable rate of decay. Now, carbon-14 is an atom that has more neutrons in its nucleus. It was one of the first atoms used to do isotope dating, and it's why it's still used today. We've got a predictable rate of decay. We know that while that organism was living, it was fixing or putting carbon-14 into its body, and that when we dig the fossil up, we can see where the carbon-14 has decayed to, know what it should have been, and then that gives us the more accurate age of that fossil. Now, carbon-14 is, at least now, fortunately for us, one of the cheaper ways to date fossils. There are much more sophisticated isotopes that can be used. And so when we are reading case studies or when we're reading about the age of fossils, the assumption is that you know that this isn't just a simple carbon-14 test. At this point, scientists have many, many different isotopes, and they've got a lot more data and evidence to support the age of that fossil. The reason why we need to know that is we want to make sure we're showing the relationships between species. Who speciated and became which species? Who's closely related to other species? We want to make sure that that relationship is accurate, and the only way to ensure that is if we have a more accurate age of that fossil. So, geology is hugely helpful, not only for understanding some of those abiotic and biotic factors that are affecting us and affecting life on the planet currently, but also understanding the historical effects on life on this planet, and for helping us to age the fossils so that we can understand what life used to look like and how it is related to life that we currently have today. Now, I keep jumping ahead or talking a little bit about life and the origins of life. Now, there are a couple of theories on how the atoms could come together to make macromolecules, organic molecules. Now, when we're looking for organic molecules, we're looking for molecules that contain carbon atoms. There are 4 main organic molecules that we find in all living things: proteins, lipids, carbohydrates, and nucleic acids. Now, with all 4 of those organic molecules, we know that they have carbon atoms, and the big question that scientists are trying to figure out is, how did the atoms come together to make those proteins, lipids, carbohydrates, and nucleic acids that we find in all living things? So this is where we get to the theories on how those macromolecules could have come together and eventually made a cell. Now, we'll talk about the origin of a cell in another lecture. I think that scientists are always hungry because these theories have food-based names. So the first theory that scientists started working on was the primordial soup theory. This idea that in the bodies of water, you would have had carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. You would have had all of those atoms that have unfilled outer electron shells. They would have been bouncing into each other. They would have been making the bonds. Okay. But there's some problems with the primordial soup theory. With all of these atoms bouncing into each other, what's the probability that you would have made carbohydrates, proteins, lipids, nucleic acids? What's the probability that you would have made those 4 macromolecules? The ocean is so dilute. So with the primordial soup theory, it hung around for a while, but it never really sat right with scientists. That they couldn't really address this probability issue. So that's when I started thinking about catalysts. Well, what types of things act like catalysts to facilitate atom reactions and molecular interactions? What types of things would help speed up these molecular and atom level reactions? Well, this is where they started looking at the crust. Well, if we look at the crust of our planet, there are a lot of atoms and molecules in our crust that act like catalysts, that facilitate all kinds of interactions. Well, this is where we get the primordial pizza theory. You've got some kind of base, some kind of crust, and all your atoms are interacting together and they start to make those proteins, lipids, carbohydrates, and nucleic acids. Well, okay, but there was still a probability issue here, and that was one of the main critiques of the primordial pizza theory. Next up is the primordial sandwich theory. And the reason why we've got the primordial sandwich theory is because we need to increase the probability of atom and molecular interactions. So when scientists started looking in the ocean, they find these huge sheets of mica. And these sheets of mica are very thin. There's a lot of surface area. A lot of surface area means a lot of space for interaction. The swashing back and forth of the ocean, all of that turbulence, the heating day and night and cooling, all of this would have helped facilitate the reactions. Now with the mica sheets, essentially being the sandwich, the bread of the sandwich, we get increased probability of atoms bumping into each other, increased probability of making proteins, lipids, carbohydrates, and nucleic acids. Now, these theories have been developed alongside some scientists who are actually trying to recreate those early atmospheres and see what they could make, see if they could make proteins, lipids, carbohydrates, and nucleic acids. And this is why it's important to take a look at Stanley Miller's work. With Stanley Miller's work, he started with this sort of simplified early Earth atmosphere. Put in some methane, put in some water, add a little electrical zap, and what do you get? You get proteins. He got 11 of the 20 amino acid monomers, and he got urea. Urea is a byproduct of larger protein molecules breaking down. So in his relatively simple experiment, he was able to make some of those building blocks of a cell, building blocks of life. This got everybody super excited. And there are still scientists today who are trying to mimic those early Earth conditions to show us and to give us evidence on how the molecules could have come together, how we could eventually make a cell, how we could make life. So we're going to take for granted that those molecules came together and that we finally have cells, because we want to look at how life has changed. We don't want to stay focused on just the atoms bonding to make molecules stage. Before we can get to a cell, I did want to focus on one of the kind of crucial aspects of life on this planet. This idea that information can be passed from one generation to the next. That information is in the form of a DNA molecule. So the DNA molecule has genes. Those genes are read to make something called messenger RNA, and the messenger RNA is used to make the proteins. The proteins give us our traits. Proteins like the keratin here in my hair, and this keratin happens to have a red color. Now you may have a different colored hair. We know that in humans there are a lot of different types of hair color. It's all based on the keratin protein, based on the genes, some of the genes that are found on chromosome 19 in our cells. So we look at how life could have originated on this planet. There was a question of what do you need first? Do you need the protein, like keratin? Do you need that in-between molecule, RNA? Or do you need the gene? Which of those is actually the most important to making a living organism? So a lot of people jumped at the idea of like, well, it's got to be the DNA molecule, right? I mean, the DNA molecule has the genes. If you don't have the genes, you can't make the mRNA, you can't make the protein. The DNA molecule controls everything that's going on. Other people are like, well, I don't know. Maybe it's a protein. But you can't actually make a protein. You can't put together the sequence of amino acid monomers, those smaller chunks, without first having the coding. But you can't go straight from the DNA to the protein. You have to go DNA, mRNA, then protein. So maybe it's that mRNA that's the most important. That RNA molecule ends up being the most important. With an RNA molecule, it's just a single strand. We can use that to make proteins. But then we can also use the RNA molecule to back calculate and make the DNA gene. RNA molecules are hugely flexible. They can act like an enzyme, breaking things apart and putting things together inside of a cell. The RNA molecule is far more flexible. And it's a lot simpler than the double helix DNA. It's a lot simpler than a protein. So, when we look at which molecule would have needed to come together first, we needed the atoms to bond together to make RNA. Then from the RNA, we can make the DNA, we can make the proteins. We can start to make some of those crucial molecules inside of a cell. We can start to see the beginnings of life. So that's where we're at as a scientific community, is that we needed the RNA molecule to come together first. Now I found a phylogenetic tree that encapsulates this, and that's in the lecture slides where we see all the possible RNA molecules, how they could have come together and eventually given us a cell to give us bacterial cells, archaea cells, and eukaryotic cells. Those three main domains of all life on this planet. One thing to keep in mind, whether we're looking at the four organic molecules, proteins, lipids, carbohydrates, nucleic acids, whether we're looking at the origins of life, whether we're looking at RNA molecules, proteins, DNA molecules, the one thing to keep in mind is that any unsuccessful combination would not still exist. We have those same forces of differential survival acting on molecules as they are also acting on life on this planet. Anything that is not successful is not likely to get passed on, not likely to persist over time. So all we are witnessing is the successes. We don't necessarily get to see the failures. And therein lies a frustration. Because in the failures, we could see all of the possible attempts, but we don't get to see those anymore. All we've got is a successful result. So keep in mind, as we're moving throughout the rest of the semester, we are only going to be making observations of the successes. We don't often get to see all the boo-boos, all of the mistakes. And I know that in life we are taught that mistakes are bad and nobody wants to make mistakes. But the reality is that there is often more information in a mistake than there is in a success. So in evolutionary biology, We always want to look for where the mistakes are, knowing that we may never find those boo-boos or attempts, first attempts, second attempts, and maybe all we get to see is the success. As always, if you have any questions, please, please, please make sure to email me or drop into Zoom office hours.